Welcome everyone to the Watchlist, Australia's newest investor webinar series for ASX listed companies to pitch to real investors and hopefully be added to their watch lists. I know each of you carefully curate your own investment watch list. Some of the companies on there you have invested in, and some of the companies are on there just so you can keep an eye out for them and potentially look at investing in the future. For companies, getting on your watch list is a big deal. It means they're being watched and have the chance to have you invest in them and follow their journey. Today, we bring together another cracking group of companies for you to consider adding to your watch list. Presenting today are EMVision Medical Devices, Orthocell Limited, Argenica Therapeutics Limited, and Arovella Therapeutics Limited. Each company will present, then management will be on hand to answer any questions you may have. So make sure you have questions ready of management and submit them using the Q&A box at the bottom of your screen. It's a rare opportunity to talk directly to management. Okay, so let's get started. Our first presenter is Scott Kirkland from EMVision Medical Devices, ASX code EMV. EMVision's mission is to be a leading global medical imaging company with its EMVision brain scanner, the product of over a decade of research and development at the University of Queensland. We are going to do things a little bit different today. This isn't a standard presentation. Scott is going to take you directly into the technology, show you how it works, and let you understand it in first person. So, Scott, over to you, and I look forward to the presentation.
Thanks, David. Much appreciated. So I'm MD, CEO of EMVision, also a co-founder. For those that aren't familiar, we're now headquartered in Macquarie Park, and we have a small team based out of the Translational Research Institute in Brisbane. Might try and, David, if we could pin my video, that would be helpful. I don't know if that's working. Hopefully people can get a good look. All right, so we are developing two products to reduce the global and societal burden of stroke. Our first product here is our Generation 1 device. It's a cart-based brain scanner designed for in-hospital use. I'll give you a little bit of a run-through of how this device works and its value proposition shortly. And then we also have our second generation helmet model designed for road and air ambulance. And what we're focusing on is to provide rapid brain imaging insights in environments and settings where clinicians need urgent brain imaging. There's a time-sensitive medical emergency like stroke, but there is limited access or no access to CT or MRI. And those two machines really are the standard of care today in brain imaging. CT came out around the 70s, MR 80s, and fantastic images as we all know, but very large, complex machines, a lot of infrastructure requirements, a lot of expense, and really not widely available at the point of care. Even the mobile versions are still 600 odd kilos and driven like a forklift. So that's the real unmet need that we're seeking to solve. Environments where we need brain imaging, but there's nothing available at hand. Clinicians are restricted to observing the patient, questionnaires, exams, looking at their pupils, these kinds of things. So focusing on stroke. So the reason we're focusing on stroke is it is a huge societal burden. So the stats are really alarming. It's 1 in 4 adults will have a stroke in their lifetime. 2 out of 3 result in some form of permanent disability. The health economic impact is also huge. The burden globally, it's around 34% of all global healthcare expenditure is related to stroke. So that's both the upfront direct costs of care and looking after that patient initially, but long-term rehabilitation, assisted living care, and the like downstream. So huge burden, very effective interventions and treatments, incredibly time sensitive. So time is brain is the common theme in stroke, and it's just been National Stroke Week. So it's all about identifying symptoms of a stroke as quickly as possible, getting that individual to urgent stroke care, brain imaging, and the right centre for treatment and intervention if they're eligible. So here's our first generation device. I'll give you a quick run through on how it works. We have 3 of these devices currently at leading comprehensive stroke centres in Australia as part of our clinical trials, so Liverpool Hospital, Royal Melbourne, and Princess Alexandra, and I'll talk a little bit about the trials in a minute. But one of the key value propositions of the device is it's a lot like ultrasound. So it's on a cart, it's portable, it's easy to maneuver, And it doesn't require all this custom infrastructure and shielding that you do with a CT or an MRI. So this is all self-shielded, past DNC testing. It doesn't interfere with other equipment in the wards, in hospital environments, and that equipment doesn't interfere with our signals either. So we have a ring of antennas in our headset that send and transmit and receive signals. One sends, the rest listen, and that process happens in milliseconds. Our headset is designed to fit most of the adult population. So from 5th percentile, smallest female, to 97th percentile, largest male. We have a silicon membrane that inflates and deflates to accommodate different head sizes. And within that membrane, there is a coupling media. It's a gel-like substance that helps our signals penetrate into the brain. Now, like most medical devices, if there is an attractive potential consumable revenue stream, you want to pursue it. And we have two consumables, the first of which is an infection prevention cap. So that just plugs onto the headset to minimize any risk of infection or eradicate. And then the coupling media itself is replaced on a monthly basis. And we're targeting approximately $25 per scan between the two consumables, and approximately 150,000 US for the device itself. So I'll run through a demonstration scan with this phantom head, and what you'll see is it will inflate. So I should mention it's operated at the side of the bed. Because of the safety profile, non-ionizing, it doesn't require a specialist operator or radiographer. So any healthcare professional with the appropriate amount of training can operate our device. So in our trials at the moment, nurses, for instance, are doing a significant volume of the scans. And so what you'll hear now, you may hear hopefully, is the fluidic system is pumping the membrane up to fill with that coupling media, which effectively removes the gap between our antennas and the head. It's as if the antennas are flush on the head. And I've been scanned quite a few times, and it's relaxing. I joke it's a bit like a scalp massage at the barbershop. It's not in any way claustrophobic like an MRI can be for many people, and is non-invasive. So once the system is fully inflated, the scan itself is very, very quick. We'll run through the scan now. And what it will do is it will output an example image from one of our earlier studies that we published out of the Princess Alexandra Hospital. That was a early successful proof of concept study with 50 confirmed strokes. So that's the length of the scan cycle. From moving it to the bedside, doing the whole workflow, performing the scan, we're targeting under 10 minutes at a minimum. We do plan for a preventative maintenance and service contracts as part of the revenue model. They typically represent about 10% of the capital equipment per annum. What we also have is 2 scan positions to cover the entire brain. In our earlier prototype, there was a solitary scan position. You can see the liquid. It's going back through the fluidic system into the container. And hopefully there might be a bit of glare on the screen, but hopefully you can see the output. If not, on the ASX, you'll see our past presentations with examples of the images. And what we're doing is anatomical reconstructions of the brain, so inner, outer boundary, grey matter, white matter, ventricles, and then we have a separate series of algorithms that will determine if a stroke is present, so is stroke or not. If there is a stroke, is it ischemic or hemorrhagic? If it is ischemic, is it potentially a large vessel occlusion? Where is it in the brain? So localizing it as well and show a picture. And it's important to appreciate, I mentioned we're not trying to replace a CT when there's a CT in the room, but and likewise with an MRI. There's always a trade-off, but what we are expecting are images that are of suitable quality, good enough quality to drive transfer, triage, or potentially treatment or intervention decision at the point of care when there's limited access to other modalities. So it's an additional tool in the toolkit that's portable, affordable, easy to operate, and fills a gap in environments where there is nothing today. So that's our Gen 1. In terms of where we are at the moment, we're running a multi-center clinical trial. We have 2 phases to this trial: a pre-validation, which is the one we're running at the moment, and then a validation phase. The objectives of the current phase, in no particular order, are safety, hardware verification, usability, and more data to feed into our AI algorithms. And then the subsequent phase is a validation phase to confirm sensitivity, specificity, and we intend to take that data to the major regulators, FDA, etc. At the moment, in stage 2 of our study, we are enrolling 150 suspected strokes. So these are patients presenting to the emergency department with stroke-like symptoms, and they will be assessed. If suitable, they'll be enrolled. They get a standard of care, so a CT, for example, and then we get a scan immediately afterwards for intervention, for treatment, but not slowing down the process. And then if there are follow-up scans, day 2, day 5, etc., follow-up CT, follow-up MR, we'll also get a scan then. And 150 suspected strokes, so some of these patients will end up being mimics, so patients that come in with migraines, epilepsy, rare cases brain tumours, and approximately half may be confirmed ischemic or hemorrhagic stroke. So we're getting data to support the application of our device up front in, for example, rural and regional settings where there is limited CT access to inform those really important transfer triage decisions and potentially treatment decisions, but also follow-up scans to help us understand response to treatment, complications. There's a whole host of things that can go wrong once a patient is in ICU or in the stroke ward. And it's very hard to move them down to radiology. It can take a few hours. Quite a few team members are involved in that transport process. And so if you can routinely monitor it, these patients, particularly high-risk patients, there's a good chance we can pick up complications much earlier and intervene earlier to reduce their length of stay in ICU. So that's all critical information. We have a fantastic clinical advisory board supporting us. We've got excellent support from the Australian Stroke Alliance, funded through the Medical Research Future Fund. Our team itself, so the senior leadership team, is made up of Stuart Crozier, who's our Chief Scientific Officer. He previously licensed IP to Siemens and GE that's now in most MRs, so he's got a fantastic track record in medical imaging innovation. Farouk Cannon is our Chief Technology Officer, incredibly experienced, managed a team of around 60 engineers at Nanosonics to get the Trophon to market. Christian Wright is our Head of Regulatory. He's got over 40, 50 FDA approvals under his belt. Robert Tiller heads up our design and is the founder of Tiller Design, a leading industrial design business. So we've got a fantastic team to get us through the process. In terms of the reimbursement landscape in the US, we're exploring things like the new technology add-on payment, which is a potentially attractive option for innovative technologies that are new to market. In parallel with all this clinical trial activity with our Gen 1 device, we have quite a few milestones coming up with our Gen 2, our helmet system. At the end of this quarter, we have an advanced prototype fabrication, and then moving into healthy volunteer studies with that device. And then next year, in partnership with the Australian Stroke Alliance, we are slated to run our first road and air ambulance studies with our helmet model. So we have 2 programs running in parallel. We're fortunate in that the team that have been responsible for developing the Gen 1 device and getting it to a production equivalent hardware have been able to be reallocated to our Gen 2 device now, and we haven't had to go out and hire a whole bunch of additional engineers to get this work done. So we operate on quite a low cash burn. And then looking forward into the go-to market for our product, we're going to be in Chicago in November at RSNA, which is the world's largest medical imaging conference. We're exhibiting there with a long-time collaborator of ours, Keysight Technologies. They're a US-based technology company, capped at about $30 billion, listed on the New York Stock Exchange. We've got a fantastic relationship with Keysight, and RSNA will be showcasing our devices. And it's, there's, I think it's over 24,000 attendees, radiologists, key opinion leaders, as well as a good place to engage with some of the large medical imaging OEMs as well. And in parallel with running our trials and getting through this regulatory approvals process, we're doing a lot of work to set up our go-to-market framework. And we do expect to use distributors in places like the US. So David, I've been talking a lot. I might throw to some Q&A if you've had some come in.
Yeah, that'd be great. Thanks for that very insightful presentation. I think a lot of people watching today would not have seen how the technology works, understand the simplicity of it and the benefits of it from a clinical point of view. So thank you for giving us that insight. Now, we do have a number of questions that have come through. Just talk us through the benefits of going through the clinical trial and the approvals process for the Gen 1 device. What will that do in terms of speeding up the process of getting Gen 2 to market? Yeah,
sure. So there's two sides to it. The first is the data that we're getting with our Gen 1 device at the moment supports the pre-hospital applications of Gen 2. Because we're getting suspected stroke patients, very acute strokes coming into the emergency department, it's the same type of patients that we'd be seeing with our Gen 2. So in terms of verifying and validating the underlying capabilities of our technology with that cohort, the Gen 1 nicely does that for Gen 2. From a regulatory perspective, it also becomes a predicate device. So we're going down the de novo pathway with the FDA at the moment. So this is for low to moderate risk devices, class 2, class 2s like ultrasound, for example. And we're proving safety and performance from ground principles. And once we have that proved out, our follow-on products like our Gen 2, there's an opportunity there for them to pursue the 510K pathway, which is an expedited pathway to market.
And what about IP protection?
Sure. So we've got, it's around 13, 14 patent families across hardware, software, calibration techniques in various stages. There's some in quite a few international, with international filings now. There are quite a few trade secrets as well that we will remain as such. So, for example, the formula for our coupling media and certain techniques that this is really really complex technology. I showed the operation of the device, which is quite simple, but the underlying technology is quite niche. There's not a lot of groups worldwide that have been working on this, and it's very, very hard to replicate. And there's certain trade secrets without which it would be close to impossible to replicate the kind of outputs that we can generate.
Now, one interesting question that's come through in terms of manufacturing the product. Have you settled on the manufacturing path and location?
So all these devices you can see here that have been built for our clinical trials have been done so in Macquarie Park. And we were fortunate to receive a $5 million Modern Manufacturing Initiative grant a couple of years ago. That's helped us get through our first production run and through to low volume production. So we have suppliers all over the world: Japan, US, China, and everything's assembled here. As we scale up, we certainly intend to keep that manufacture local in Australia, and hopefully we can pursue some further government initiatives to achieve that.
And do you have any other imaging applications beyond stroke?
Yeah, sure. So with the two brain scanner devices, we have ambitions to expand the indication out to traumatic brain injury. So effectively, we're talking about the same hardware, but a software upgrade to expand into that indication. We would need to run a smaller study to also prove out our capabilities with that specific cohort of patients. But there's significant interest from clinicians to look at traumatic brain injury and swelling in the brain. Every time we meet with our clinical advisory group, there's dozens of different use cases where they talk about a significant need for point of care imaging, but with nothing available. So it's just a matter of getting our stroke product into market, and then we can expand on our indications from there.
A good question just come through from one of the viewers. In terms of your most recent ASX announcement, you talked about the fact that by the end of August, you should have or appear to have had 50% of the clinical trials conducted. When do you think you'll be starting to put that information out to market in terms of the outcomes of those trials?
So we've said that by next quarter, we expect to complete the enrolment of 150 patients. And our strategy generally in terms of presenting insights, findings, results of our study is really all around domestic and international podium opportunities. So major conferences that are well attended, that's really our strategy to get our results and findings out.
And there's a direct question here that probably makes more sense to you than it does to me. Any updates in the latest CAA Congress?
CAA, yeah. So So for those that might not be as familiar, so the CAA is the Council of Ambulance Authorities, industry body. We exhibited recently, a couple of weeks ago. It's a really good avenue to connect with leaders in the paramedic field, in the pre-hospital setting. Some of our colleagues from Titan Telehealth were there as well. Again, you know, the common theme is when can we get our hands on this? But from our perspective as well, we're interested in broadening out our user groups to get feedback on the device, particularly the helmet device. And Robert Tiller, our head of design, has been spending a lot of time with different paramedic groups to go through the workflow, get their feedback on deployment, because ultimately we want them to be able to carry it in a backpack, take it into a patient's home, scan the patient there, and then send the information via telehealth. And you know, we know in ambulances, real estate is really limited. You know, they joke they count every bandage. So going through the workflow, where the device lives in the service centres, how it's stored, how it's manoeuvred, all those things are critical to ensure that we can get as many ambulances to adopt the product as possible.
And just lastly, obviously this is an investor-focused webinar. From a cap structure point of view, are there any names on the register or cap structure positives that investors should be aware of?
Sure. So we've a low number of shares on issue, and we've kept it like that since our existence, with very minimal dilution. We've been able to do that on the back of approximately 20 million in non-dilutive grant funds that we've secured since inception. In terms of the register, founders, management directors own approximately 20%. So everyone has skin in the game. All our employees are shareholders as well. So we're all motivated to make this as successful as we can. We have a couple of institutions in the top 10 and some family offices as well that have been very, very loyal since early on.
Thanks, Scott. A great presentation. Clearly a world-class technology, a company to watch, which we will do with interest. Look forward to following the company and its progress.
Thanks, David.
I'd now like to introduce Dr. Liz Dallimore from Argenica Therapeutics Limited, ASX code AGN. Argenica is developing novel neuroprotective therapeutics to reduce brain damage after stroke, and importantly, is entering a key period of study activity. Liz, over to you.
Thanks so much, David. Bit of a stroke theme going on today. That's great to see. So thanks everyone for joining me. I'll just give you a bit of a flavour of Argenica. We've only got 10 minutes, so it's quite a tight turnaround. So really want to focus on the stroke application and our phase 2 trial coming up. So if we just scroll through the slides, please, David, disclaimer. So this really just paints sort of a picture of who we are as a company. So what we're looking to do is to develop novel drugs or novel therapies, essentially, that are able to protect brain cells following brain injury. So the lead indication that we're going after is in ischemic stroke, and I'll come back to that a little bit in terms of how the drug works. But we really are now starting to generate some really exciting preclinical data on other brain injuries, such as traumatic brain injury and other neurodegenerative conditions. So with a kind of key focus on Alzheimer's and looking now to move into Parkinson's disease. So in going after these indications, we're going after some pretty large unmet medical needs. So currently there's no drug on the market that is able to protect brain cells following injury, such as after a stroke, or drugs that have this multiple mechanism of action, such as our lead drug candidate, that can sort of protect brain cells in a number of different ways. And that multiple mechanism of action is really important when we're looking at neurodegenerative conditions. So as we start moving into our phase 2 clinical trial, what's fantastic from a company point of view is we're actually starting now to get some really keen interest from pharma companies to see what we're doing. So the company itself, we did list just a little over 2 years ago, so in June 2021, and in that time we've kicked some pretty big goals. So we were a preclinical company when we listed, and now we're about to start a phase 2 clinical trial. So 2 years is a pretty small time frame to move a company from preclinical to phase 2 clinical trial. So we're really proud of the work that we've done, securing our manufacturing, undertaking all the preclinical studies to get into the clinic, successfully completing a phase 1 clinical trial to show that our drug is safe, it's well tolerated in healthy human volunteers, and built that really fantastic clinical and reg team around us. So in doing all that, hitting all the milestones that we said that we were going to do, and probably accelerating some of them even, we've built some really significant value for shareholders. So next slide, please. So just quickly on some of the key company metrics. So pretty well funded at the moment, and we've got a R&D tax rebate coming through likely next quarter. What's fantastic is in the last 12 months, we've been able to secure just a little over $4 million of non-dilutive funding. And that is really to fund those preclinical, those other indications beyond stroke, so that we can conserve cash that we've raised really to progress that phase 2 clinical trial. Next slide, please. So our lead drug candidate is known as ARG-007. I won't go into too much detail here, but some of the key points to pick up from this slide is that this is not I guess, an overnight type of flash in the pan sort of company, even though we've only been listed for 2 years. The drug development has been going on for a little over 10 years. So the drug itself has spun out of the University of Western Australia and the Para Neuroscience Research Institute here in Perth, where I am. It's had over 25 peer-reviewed publications. That's mainly in ischemic stroke, but also efficacy data showing that the drug is working in traumatic brain injury, that it's working in hypoxic ischemic encephalopathy in infants, and Alzheimer's disease as well. We do own all the patents as the company, and we're free of any encumbrances back to the founding research institutes. And as I said before, we've managed to significantly de-risk this drug in showing that it is safe and it is well tolerated in healthy humans. So that's a really pivotal milestone for the company that we've now achieved. Next slide, please. So just quickly, if I go into, I guess, ischemic stroke, I'm not sure sort of what Scott's covered. Apologies, I was late jumping on, but in terms of an ischemic stroke, when someone has a stroke, and if we look at the 2 different types of strokes, so we've got the ischemic stroke caused by a clot in the blood vessel in the brain, that makes up around 85% of all strokes. That really is the type of stroke that we're focused on in this drug. That's where all our preclinical data lies. So when you have a clot in the brain and it prevents the blood flow going to that part of the brain cells start to die because the blood flow carries oxygen, and brain cells can only last about 3 seconds without oxygen. And because brain cells all talk to each other, as soon as you get that blockage in blood flow, that reduction in oxygen, you get this proliferation out or cascading out of brain cell death. And around 1.9 million neurons are dying every minute that treatment is delayed. So what our drug is aiming to do is to stop that cascading out of neuronal cell death. So essentially hibernating those brain cells and buying that patient time to get to hospital. Next slide, please. So if we just look at that clinical workflow, so currently when a paramedic arrives, there's very little that they can do in terms of triage. So the name of the game really is to get that patient to hospital. So the main treatments for ischemic stroke, around 9% of patients are eligible to receive a clot dissolving drug known as TPA or TNK. That dissolves the clot. But the type of treatment now that's really starting to take off is a mechanical removal of the clot, known as a procedure known as thrombectomy. So this is where a radiologist will go in and actually mechanically remove the clot. But we really need to diagnose that, the type of stroke, where that clot is, etc., to work out the treatment. So in that delay of getting that patient to hospital to either get a thrombectomy or to receive the TPA, you get this increasing amounts of cell death. So we're aiming to ideally be able to deliver our drug as soon as possible after stroke onset, and that will suppress that neuronal cell death, reduce it, and buy that patient more time to get to the hospital and improve those functional outcomes. Next slide, please. So as I said, we've got huge amounts of preclinical data showing that our drug really is working really consistently in these preclinical models. This data is from a non-human primate study that has been published and showing that our drug is able to reduce the total amount of brain cell death by 66% following a stroke and following restoration of that blood flow. So really mimicking that clinical setting that you see in humans. But what's really great about this data is that it also shows that that reduction in brain cell death, you still see it 28 days post-stroke. So you do get this secondary wave of brain cell death that happens after the clot is removed. So what this data is showing is that our drug is still protecting against that secondary wave of brain cell death, which is really important now that we're translating that into the clinical setting. Next slide, please. So what do we know? What have we sort of done to de-risk this drug? Why are we really excited as we move into this phase 2 trial? We know that in all the preclinical models, it's significantly reducing that brain cell death. We know that it doesn't exacerbate bleeding in a hemorrhagic stroke model, so it would be safe to deliver in the field, in ambulance. We haven't seen any adverse events in our phase 1 healthy volunteer study. And we also know that it can be delivered with standard of care, that clot dissolving drug. So really important in drug development to make sure that your drug is not having any adverse effects on current drugs being used or vice versa. Next slide, please. So if I just quickly touch on the phase 2 clinical trial. So we have submitted our ethics application seeking approval to undertake this trial. We expect to get that, hopefully that approval in the next couple of weeks. And that is a significant milestone for the company, getting that ethics approval and being able to now start our pivotal phase 2 trial. That trial will be done only in Australia. So at the moment, we're onboarding 9 hospitals. We expect to onboard another hospital in the coming months. So when someone comes into the emergency department, they will be scanned. We'll determine the type of stroke that they've had. And the reason that we're determining the type of stroke is that we want to limit this trial to strokes or clots that are only in large vessels in the brain. And that's just really good clinical trial design. It essentially reduces the amount of variability that we would see in the brain arising from that type of stroke. So it really is a safety trial. We want to show that the drug is safe, it's well tolerated in ischemic stroke patients, but we will also be looking at a number of efficacy endpoints so that total volume of brain cell death reduction will be one of the key endpoints that we'll look at, but we'll also have a number of functional assessments. So see how those patients are going after receiving our drug. So in terms of time frame, I've just got some key milestones that we're aiming to hit over the next 6 months, but really that first patient dosed should be early Q1 at next calendar year. Next slide, please. So I'm probably running out of time a little bit here, but I do want to touch on the pipeline of additional indications that we have going on in the company, because this is where I'm really excited about this preclinical data that's starting to come out of these other indications. Because our drug works on multiple mechanisms of action, it's able to protect brain cells in a number of different ways. It lends itself to being effective in a raft of different neurological conditions. So we've announced earlier this year that our drug is working on protecting brain cells after a moderate traumatic brain injury. So that would be akin to a pretty severe hit in a sporting context or a king hit that you might get. And so showing that our drug is protecting those brain cells, those neurons from that injury, it's reducing inflammation. And we've now been awarded a federal government grant to progress that work into a more mild traumatic brain injury akin to concussion. We've also got some really exciting data that shows that the drug is reducing protein aggregation, so can lend itself to neurological conditions such as Alzheimer's disease, Parkinson's disease. So this is absolutely, we are really excited about this application. There isn't a drug out there at the moment that's able to reduce multiple protein aggregates at the same time. Most drugs only work on a single protein aggregate, so only beta amyloid, only alpha synuclein, only tau. So really excited to continue to explore the potential of our drug in those neurodegenerative diseases. And then hypoxic ischemic encephalopathy, or HIE. This is a rare pediatric condition. It usually happens during labour as a result of, say, the cord around the neck or preeclampsia, where the baby is exposed to a significant reduction in blood flow, which results in huge amounts of brain cell death, can lead to cerebral palsy. Again, awarded some amazing funding from the Stan Perrin Charitable Foundation to do all the studies that we need to do to progress that, our drug into clinical studies in HIE. Next slide, please. Yeah, so look, I'll just wrap up. I'm sure I've gone over time, but really, we're really at a pivotal moment for the company. So starting now to generate proof of concept data that our drug is actually working in humans in ischemic stroke indication. So that's where now we can start having discussions with pharmaceutical companies, getting them across the data and looking at potential deals post coming out of that phase 2 data readout. We are then also progressing all these other indications. So the drug seems to be really multifaceted. There's multiple mechanisms of action lending itself to a number of different indications, which is really, really exciting for the company to start progressing that. So really exciting to be part of this company. The next sort of 18 months is going to be really pivotal, and we can't wait to get started. So thanks very much for your time.
Thanks, Liz. Great presentation. Clearly a very important market that you're playing in, and a lot of people will know of or have been touched by people who've had strokes. So anything that can assist in the regeneration or the improvement of those outcomes is very much a focus for a lot of people. Now, one question, taking back a little bit, your own experience getting to this point, was this an area you were always focused on, or have you been led to this path through the other work you've done and from a clinical point of view?
In terms of me personally, or okay, sure. So I guess I am a neuroscientist by training. I did a PhD in neuroscience looking at neuroplasticity following trauma, so the ability for the brain to change itself. Have worked in stroke research, so I worked with the inventor of this drug 25 years ago. So always, myself personally, have always had a passion for neuroscience. I absolutely love it, and we are on the brink of, I think, a kind of a neuroscience revolution and a renaissance for pharma companies to start getting some of these drugs approved. So I did, post PhD, go down the business path. So it's very nice to be back in the fold of combining the two. So yeah, loving it.
And you touched on at the beginning that pre-listing, a lot of work has gone into this drug development. This isn't something that's new in terms of the work and the amount of work that's been done. This is a project and a development that's been occurring over a long period of time. Yeah,
absolutely. So, I often hear people say, drug development is such a long path, and it's sort of a hard investment thing to come into. But the point that I make is that, this drug has been around for 10 years and it has been significantly de-risked. We've got 13 peer-reviewed publications in all sorts of different models looking at stroke to show that every single time this drug is working in those models. So huge amount of de-risking work that's gone on. And now we are now at the cusp of showing that the drug works in humans. So we're only sort of 18 months, 2 years away from that. So this is the time that, I would say get in, because we're still pretty cheap. But, there's a lot that's going to happen in the next couple of years. So it's exciting.
From an analogy point of view, when a drug's developed, and are there any examples, I suppose, that you can think of a drug that is moving through the phase you're moving through to, and what 18 months later looked like from a value to the shareholders? I might be putting you on the spot, so I apologise.
Yeah. Look, I think it's always a little bit hard to compare different drugs, because there's different indications, there's different nuances to, I guess, to sort of the deals. I do often get asked about Neuron. So they obviously had a big value inflection point post phase 3, but they did manage to do a deal with Arcadia post their phase 2 readout. Possibly not the deal that the shareholders want, but what I do often say to people is that in what they've got to realise, in Arcadia was taking on a huge amount of risk in that Rett syndrome. Phase 3 trial had never been done in that indication. So that's, looking forward, it is actually a really good deal. So I guess it's just about how you sort of communicate those sorts of deals. So any kind of partnering you can do with a pharma company to help you progress through into phase 3 is always going to be a good thing.
Now, I am going to put you on the spot again, and I know this nothing's been announced, so this is just a conjecture. When does Big Pharma get interested? When does Big Pharma go, hang on, I've been tracking this for X years, now I'm interested because they've de-risked it?
So we often get... I like to think pharma starts coming to us. So once you get that tipping point where you're not going out so much sort of promoting, you're actually having people come to you and say, hey, look, we've been following this. We understand that you're sort of at this point. That's always a good sign that they're, that you're on their radar. They do typically want to see that proof of concept in humans in a particular disease indication. So getting that sort of phase 2 data readout, where we're at though at the moment, because we have so much data on this drug, having those conversations around getting some early stage due diligence, that's sort of, we're definitely at that point at the moment.
Just to remind everyone, nothing's been announced, and if materiality was there to be announced, it would be, but clearly it's good to hear that conversations are happening, and as they should be with a company in your stage. Last question, post phase 2, what are the steps to commercialization?
So commercialization is a bit of a funny one in drug development, because you could look at a deal as commercialization. So that could be post phase 2, and we're open to all sorts of deals that are going to provide shareholders with the biggest bang for their buck. But in terms of that clinical development pathway, the next stage is to do a phase 3 clinical trial, and that's a big multi-centre global clinical trial. So we do really want to partner with someone in terms of executing that trial. The time frame for that trial isn't necessarily dissimilar from a phase 2. It's just you have to onboard a lot more sites globally. So timeframes, it could be potentially done in 2 to 3 years, depending on how many sites you onboard. Obviously, the more sites you onboard, the more it costs. So it's sort of finding that balance between cost and time.
Well, as you say, targeting large addressable markets, a lot of work's been done that set the company up beautifully for the next 12 to 18 months. Exciting times ahead. We really look forward to following the progress. Liz, thanks for your time.
Thanks, David.
Now, I'd like to introduce Dr. Michael Baker from Arovella Therapeutics Limited, ASX code ALA. Arovella, a biotechnology company, is focused on developing therapies to treat human disease. Its 2 focus areas are oncology and conditions that impact the central nervous system. Michael, we look forward to hearing more about it. Over to you.
Wonderful. Thanks, David. Thanks very much for the introduction, and thank you to everybody that's put this webinar together. Thanks, everybody, for taking the time to dial in and listen. And I'm excited to tell you about Arovella Therapeutics. As David just mentioned, we are a biotechnology company now, pretty much focused on oncology. And for those of you that don't know us, our ticker is ALA. Next slide, please. and I will be making forward-looking statements, so please do take note of the disclaimer. And just jumping into the highlights of the company. So what we're working on is termed an invariant natural killer T cell, or iNKT cell for short, and it's a cell therapy platform that we're developing for the treatment of initially blood cancers, but looking to expand also into solid tumors. And I'll unpack what that means more in a moment. Our lead product is termed ELA-101, and it's being developed for what we call CD19 expressing blood cancers or lymphomas. And we're expecting to take that into clinic in 2024, which will be an exciting milestone for the company. Now, the cell therapy sector has been a really exciting space to consider the next pillar of oncology therapeutics, but it has been hampered by some challenges. And we believe that our iNKT cell platform is well positioned to help tackle some of those challenges and push the whole field forward. Now, we've developed a very strong leadership group with a lot of experience right the way from the research and development phase all the way through to commercialization. And there's a heavy emphasis within the team to develop cell therapies, which is quite specific and unique. In terms of the company's DNA, we have essentially worked hard to pivot the company in doing so, bringing in what we consider to be the most high likelihood of success IP from a range of institutions and universities worldwide. So we've got our lead platform from Imperial College London, and we've since acquired technologies from the MD Anderson Cancer Center, and also have an option agreement for another technology from the University of North Carolina. And we'll continue to do that where we see that there's a good fit for some of these technologies to fit with our current platform. So we are the only iNKT cell company in Australia and one of a handful globally. So we do see that we have a unique value proposition and that we are well positioned for success. Next slide, please. So I'd like to quickly unpack some of our financials. These dates are towards the end of July. So we're somewhere in the range of 40 to 45 million in terms of our market capitalization. Our pro forma cash, including the proceeds from an SPP that was closed at the beginning of July, was about just under 7.4 million. And of course, that does not include our R&D tax rebate, which we're expecting to bring in later in the next quarter, or in this quarter, I should say. And what we're very happy with our current register. We're delighted to have Merchant Group as our largest shareholder, but also equally delighted to have a number of other smaller institutions and high net worths that are quite supportive of the company in terms of its drug development efforts. So we've had a good start to 2023 off the back of presenting some very exciting data demonstrating what we're trying to embark upon and take into clinic at the American Association for Cancer Research in April. We had quite a spike in the share price. Look, it has since come off a little bit since then, but we are still excited for some of the news flow and the milestones that we're expecting to hit towards the end of the year that we're going to continue to deliver shareholder value to all of our shareholders. Next slide, please. Now, I just want to unpack exactly what it is we do, and apologies to those of you that already know this space well. But what I'll do is I'll start with CAR T because it's the most well-known cell therapy. And where the T comes from, just very briefly, it relates to an immune cell in our bodies. It's naturally occurring all the time, and they are called T cells. And their job is to find and destroy or eliminate anything that doesn't belong, whether it be an infection, in some case, cancer cells as well. I don't have a pointer, but moving to the diagram, the way this actually works as a therapeutic for cancer treatment is we actually take the blood of the patient, and I'd like to make that very clear. It is from the patient that actually has in this case, the lymphoma. We then isolate their T cells, the immune cell. They get shipped off to a lab to be genetically reprogrammed so that they actually have, and you can see it just on the right in the middle, it's called a chimeric antigen receptor, or CAR for short, which is where we get CAR T. And I'd like you to think of that as a laser-guided missile that now will tell that immune cell to go and destroy the cancer cells that have a particular marker on their surface. So they get grown up and going in the lab into the millions or billions, and then ultimately they end up going back into that patient where they originally came from. And you can see on the surface of the cancer cell, there's little green bumps. The newly engineered T cell will find those bumps, and as soon as it does that, it will trigger the cancer cell's destruction. So just to make it clear, we start with the patient's material and we give it back to the patient. Next slide, please. So this has, as I mentioned a moment ago, it has revolutionized the way we think about cancer therapeutics because we are now at a position where we are starting to use the word cure and cancer in the same sentence. And this is off the back of a series of media reports coming out after some research demonstrated that patients that had taken the therapies both in the adult setting and the pediatric setting, they were now 10 years cancer-free. So that's last year, so it's even beyond, which is quite exciting. Accordingly, the first two approved products have done quite well in terms of revenue. And naturally, with a market like this for oncology, it is expected to exceed more than 60 billion by 2030. So it's set to be a very important pillar, both from a healthcare perspective, but also commercially. Next slide, please. Now, as I mentioned earlier, there are a number of challenges that do still hamper the therapeutic class as it stands today. And I mentioned a moment ago that T cells that are used in this process, they must actually originate from the patient. And what this means is that for every single therapeutic dose that's manufactured, it needs to be manufactured bespoke. And the pharmaceutical industry, like many industries, wasn't necessarily set up to be making a therapy for every single individual. So what this results in is quite a complex supply chain, and naturally a very high manufacturing cost, which leads to the therapy costing more than $500,000 per dose per patient. So also we know that because the starting material comes from the patient, it's likely that the immune cells themselves are compromised due to the disease or due to certain treatments like chemotherapies that can actually reduce the fitness of the immune cells. In addition, not all manufacturing or cancer centers are actually able to manufacture these types of therapies. So there will be a number of eligible patients that naturally will not be able to be treated with those particular CAR-T products. So in addition, it takes about 4 to 6 weeks to get the therapy manufactured for each patient. And unfortunately, some patients will not make that journey. They'll actually die before the therapy can be manufactured. And of course, because we're continuously making it for every single person, there's a high likelihood, well, not a high likelihood, but there's always the likelihood of a manufacturing run failure, which again increases time and costs. So we do believe that our iNKT cell platform addresses these manufacturing and logistics challenges, but we also believe it has the potential for improved efficacy over what's already available. Next slide, please. So I would like to briefly introduce the invariant natural killer T cells, or iNKT cells for short, just so I can contextualize why we're so interested in them. And what you can see in the top right corner is the T cell, which is where most or all approved CAR-T products sit. Bottom left is the NK cell, which is also quite a well-known cell type that people are using in this space. If you just click on the, click forward again, please. But as I said, we're focusing on the iNKT cell. And the reasons we're doing that are they actually are the front line of the human immune system. They bridge the two halves of our immune system, which we call the innate, the fast acting, and the adaptive, which is the more slow but prolonged response. They actually contain the ability or the killing mechanisms from both T cells and natural killer cells. And we know that they actually naturally target certain cancers that also already produce CD1D. Now, they also have a very strong safety profile. They don't cause what is referred to as graft versus host disease, which is what enables us to be able to give iNKT cells from a healthy individual to somebody that has cancer. So we can actually use these for, in one sense, a one-to-many approach, or what we like to call off the shelf. Now, in addition, unlike some of the other immune cell types, they also have multiple anti-cancer properties. They shape what we call the tumor microenvironment, which is the area surrounding the tumor, by actually influencing and killing certain other immune cells that essentially help the cancer cells to grow and evade the immune system. They also are able to actually infiltrate inside the tumors and then secrete what we call signaling molecules or cytokines that actually then activate other components of the immune system, like T cells and natural killer cells. Next slide, please. So we believe very strongly that the potential of CAR-iNKT cells is untapped. And if you could tap forward twice, that would be terrific. Once more, thank you, perfect. And this is just a snapshot of the players that are currently in the T cell and NK cell space. So I think you can appreciate that there's quite a lot of activity in each of these sectors. And as I said, this is a subset of some of these players. If we click once more, we'll see that in the iNKT cell space, there's really a handful of groups working on the invariant natural killer T cells. So we're quite excited because we see that we're in the leading edge of cancer therapeutics. in the cell therapy space, but we've also carved out a very nice position to be in a niche area on top of that as well, which is nice. Next slide, please. Now, just to give you an idea of the workflow, so I showed you what it looks like for CAR-T previously. It is quite similar. There are two major differences that I'd like to point out. So on the very top left, we collect the blood from a healthy donor. So that'll be someone with a fully functional immune system. We then isolate their iNKT cells. We take them again to a lab like we would in the previous example, where we engineer them to produce the chimeric antigen receptor or laser-guided missile, which you can see sticking up in green. And then they get grown up and expanded into the billions in our case, so that we've got many cells, then we can store them and vial them, put them in little vials, store them in the freezer. And when a patient is actually eligible to take that therapy, it can be pulled out of the freezer and then given to that patient as and when needed. So what that means in terms of advantages is we have a healthier starting material because it's coming from a healthy donor. We can scale up to have multiple doses per batch, and it's much faster access to treatment for patients because we don't have to wait for the therapy to be manufactured, and it does remove the risk of manufacturing on failure because it's sitting in a freezer waiting to be administered. Next slide, please. And so when we look at what we're actually developing, and I haven't had time to go through each of these in detail, but our lead product is ALA-101, which is targeting lymphomas. You can see on the very left-hand side, and I'll do my best to describe this without a pointer, but we've got in the middle up the top, we're combining ALA-101 or our CD19 targeting program with another well-known biotechnology company, EMVision's onCARlytics virus. That's a very neat program where their virus will actually tag solid tumors with the target that our iNKT cells will recognize. So we're excited to develop that to tackle a range of solid tumors. We did get promising in vitro data for that, or test tube data for that earlier this year, and looking forward to getting animal data before the end of this year, which is exciting for that program. And on the very right, we've got another program from the MD Anderson Cancer Center, which targets a unique target on the surface of, and the data suggests on the surface of multiple myeloma and a range of solid tumors. And in the very low dark blue line, we've got our cytokine technology from the University of North Carolina, which we think will enable us to increase the effectiveness of our iNKT cell platform across a number of different solid tumors as well. So really importantly, we can continue to look for new CARs to add to the platform, which is quite exciting, as there are a number that we can evaluate both in the blood cancer setting or the solid tumor setting. Next slide, please. So in terms of the next 12 to 18 months, we do have quite a busy schedule in front of us, which is exciting. Our major goal is to take ALA-101 into its first in human trial in 2024. Now, for those of many of you will know this already, but this will be done directly in patients. Nevertheless, this is considered a safety study, so we'll be looking to make sure that we don't see any dose-limiting toxicities or anything of that nature. But of course, we'll be looking to set that trial up so that we're looking for early efficacy signals of the product as well. And we'll also continue to assess novel complementary technologies, as I mentioned, that we believe enhance the platform and start to push us more towards the solid tumor space as well. So for the second half of this year, we'll complete process optimization and scale up to prepare for manufacturing cGMP or clinical grade material for clinical trials. One of the components, which is called a lentiviral vector, will have finished before the end of the year, which is a really important milestone for manufacturing these types of therapies, and we'll be finalizing our clinical trial plan. And so for the ALA-101 for this first half next year, the intention is to complete the manufacture of the product, phase 1 clinical trials, and then complete the regulatory submissions so that we're ready to start phase 1, which is a very exciting milestone for the company. And so in terms of our earlier pipeline programs, as I mentioned, we're working towards getting data for the combination of our ALA-101 with EMVision's onCARlytics platform in solid tumor animal models. We are analyzing additional CARs to add to the platform, and we shared some data recently about the cytokine technology that is currently under option, and pending our due diligence, we'll look to license that into the company. And so for those studies in the first half of next year, we're excited that we'll continue to generate proof of concept studies for those particular therapies. And again, looking towards moving the platform more towards solid tumors. Next slide, please. I thought this is a really useful slide just to contextualize some of the activity that's transpired in the cell therapy space. And I think we can all agree over the last 24 months, it's been a pretty tough sector in terms of capital raising, share prices and M&A activity. But what we have seen specifically in the cell therapy space is there still have been a number of, whether it be acquisitions or collaborations or strategic partnerships that have transpired. And importantly for Arovella, and I've deliberately left it at this list, but it is also in companies or technologies that are at quite early stages, early as preclinical, which is where we currently sit, but also a lot at phase 1, which is the next phase for us to enter. And some of these deals are quite sizable, in the order of hundreds of millions upfront payments. Again, putting that in context, that Arovella's current market capitalization is between 40 to 45 million. It's clear that this is still a sector where there's a lot of value to be created. And of course, looking for a strategic partner to get involved is also something that we're considering and very mindful of. Next slide, please. With that, I'd just like to say thanks again to everybody. Thanks, David, for moderating, and thank you to everybody for listening. I'd be delighted to answer any questions.
Thanks, Michael. A great presentation and clearly a sizable market you're operating in. Similar to the question I asked of Liz, is this something you've always been interested in? Has your clinical and your own research work led you to this point, or have you sort of cottoned onto it later in your career?
It's an excellent question, and my career's probably been cut up into multiple stages. So my first career was as an academic researcher, and not too dissimilar to Liz, actually, and a biochemist. And I think that really sets the foundation for these technologies. They all operate off an intimate understanding of biochemistry, how cells behave, how these things can actually be utilized or weaponized in the fight against cancer. And then I moved into drug development, followed by some time at a venture capital fund in Australia, and was very aware of the CAR T space, probably quite some time ago, probably if not somewhere around the 10-year mark ago. So it was a sector that I was always very fond of. And upon joining Arovella, I thought it was a very good idea to be looking at technologies in this space. And one of the key factors for us, and that slide I mentioned earlier, where there's a lot of activity for both T cells and NK cells, was how do you enter this and not become a me too? And I think we managed to do that by working on the invariant natural killer T cell platform. And it was quite remarkable, actually. I think a month before we licensed in this technology, another iNK T cell company was bought out for 185 million. Another one came out of stealth and raised 52 million and didn't list, but they raised 52 million in the US, and since ended up in an $875 million partnership. So it's clearly a sector, I think, that's got a lot of potential. Once we start coming out the other end of this pretty prolonged biotech downturn, I think we're going to see some strong value creation for our shareholders.
In terms of the iNKT product, what was the genesis? And apologies if you did talk to it earlier on, but where did it come from? What was the genesis? What was the driver?
Yeah, I didn't really delve into it, so it's a good question. So the technology was founded or discovered, I guess, at the Imperial College London, and that was an important part of our criteria, that we'd look really for top tier either universities or research institutes. So for those of you that don't know Imperial, it's consistently ranked the top 10 university worldwide. And when we were looking for a range of technologies, and we would have looked through, I think, close to 100 or more, this is one that really stood out to us for all the right reasons, right? That it was differentiated, it wasn't a very important sector. And importantly, the inventor himself, he'd spent his career researching and understanding iNK T cells. So that's not something that you can just sort of walk into, I think, and sort of fake. So that was really important. He's also a medical doctor, so he continues to treat cancer patients and sees firsthand the failures, what we're not getting right. And I think that was his driver to get involved. I really, firstly, in having a project that was far more translational. And then when we came and approached him to license his technology, I think there was just a really good fit and a good relationship from the outset, that we were the right group to develop his technology and take it into clinic. So again, I think we're all pulling in the same direction from that point of view, and it is a very exciting time taking this into clinic next year.
And you showed the the slide before where there was lots of people in this area and lots of people in this area, but when you looked at the iNKT cell market, it wasn't busy. Why do you think that is? And is that the opportunity, that it isn't busy? Everyone's already over in other areas focusing on other things, but there's a bigger opportunity here.
We think so. And there's a number of reasons why, as I said, the T-cell space became quite saturated. There was 2 acquisitions in 2018. One was both companies were in phase 2. One was 11.9 billion. The other one was 9 billion. And it essentially set the market on fire in terms of what could be achieved. And then natural killer cells were the next touted therapy because they can be used off the shelf or given from healthy donor to patient. T cells can't be given in that way. And so we see that as a real limitation on continuing progress. There's often media reports or fierce biotech reports that companies, the biggest pharma companies you can think of are struggling with manufacturing because it needs to be made for every single person that wants to take the therapy. Conversely, the NK cells, I think they've had trouble demonstrating the prolonged persistence in the human setting to have the durable responses that we would like to see from therapies of this nature. So that's where iNKT cells, having the good properties from both T cells and NK cells, actually fit like almost, I think another company's referred to them as the Goldilocks immune cell. Now, I think what has hampered, and we presented this recently at BioShares actually, what's hampered the uptake in the sector is they were discovered, I think 20 or 25 years plus after people were already looking at the cytotoxicity of T cells. So they have come to the fore, I guess, or to the front line quite a bit later. And it wasn't only until mid 2010s that we had the first CAR iNKT cell publications actually put out in the scientific community. So they've been a far less well-studied immune cell type. And we also know that they're lower in number in the human immune system, right? So when we actually take the blood from a healthy patient, we start with a lower amount of cells, which will make manufacturing more challenging. But what we licensed from Imperial was actually patents to manufacture the CAR iNKT cells. So I think we sit it in a very good spot. We don't know the exact reasons why the space isn't busier, but for our intents and purposes, right, we're quite happy to be in that space with relatively few competitors.
And final question, you did show a slide which showed a lot of the transactions that have been occurring in the space. Now, again, I don't want to put you on the spot, and we all understand materiality and the need to disclose at the appropriate time. But when do people start talking to you? When do people start looking and going, hang on, you're doing something that is different to what others are doing, but clearly has a significant application, albeit a little bit behind from a clinical point of view in terms of where you are in the cycle. But when do they start looking at you and going, well, we see what you're doing, we see the work that's been done, the partnerships are strong, you're moving into that phase where we now start getting interested?
I think it's now, to be honest, David, that's when you start having the discussions. And when we presented the data at AACR in April, that did generate interest, which was great for us, right? We're delighted to have that happen. But the reality is it has happened for other companies even earlier than where we are. And I gave that example before. Appia Bio came out of stealth in May 21, raised 52 million. And then by August 21, they're in an $875 million partnership with Kite Pharma. So they hadn't reached clinic at that point. They were 2.5 years away from clinic. They're still not in clinic. So, that preclinical window, it can be large. And it seems as though for the right technology, the right pharmaceutical company is willing to get involved. Naturally, I do think some of the more sizable acquisitions that have transpired, they have been in phase 2, but we've still seen a lot of activity in phase 1, which is the next step for us. So we're quite excited, I think, by what we're developing. And again, bringing in synergistic technologies that enhance our platform, and also making sure we secure IP around them and block out our competitors in the iNKT cell space. We think that just sets us up for success, and obviously sets us up to have some pretty interesting discussions as we move forward.
On that note, Michael, thanks for the presentation. We look forward to following the company with interest. Clearly a pivotal time for the company and its shareholders. We wish you all the best and look forward to catching up with you soon.
Thank you, David. Appreciate it.
Now, I must apologise. We were meant to have Paul Anderson from Orthocell Limited joining us next, but unfortunately, he has experienced technical issues that we have been trying in the background to resolve, but unfortunately have been unable to do so. So we will be pushing Paul onto a webinar presentation in the coming weeks. So he will be available shortly to present, but he is unable to present today. So apologies for that. As I said in my introduction, the purpose of this webinar series is to give you the opportunity to engage directly with companies that are on your watch list and to give you an idea of companies you might want to potentially add to your watch list so you can follow them in the future. So look at your watch list. Let us know if there are any companies on there you want to hear from, or let us know about companies you have been thinking about adding to your watch list, and we'll get them to present to you in the future. You can hear directly from them, ask the questions, and learn more about them. Thank you to the presenters for making your time available today, and to you for watching. We'll see you again in a couple